Hey guys! Today I'm going to be talking about protein synthesis. Before I start talking about protein synthesis, you need to know some background information. DNA, which is short for deoxyribonucleic acid, is made up of two strands that form to create a double helix shape. It is made up of 4 nitrogenous bases and it's located on a chromosome. A section of it is called a gene, which contains information to code for one specific protein. RNA, which is short for ribonucleic acid, has only one strand and has 4 nitrogenous bases that are different from DNA. There are 2 types of RNA involved in protein synthesis. You'll know about those later. So in short, the differences between RNA and DNA are that RNA has only one strand, it has a different sugar molecule, and it has different bases than DNA. Now we'll talk about the base pairs for DNA. Adenine is paired with thymine, and cytosine is paired with guanine. These are the 4 bases for DNA. Now the base pairs are similar to DNA for RNA. Instead of thymine though, you have uracil. I'm now going to draw a model of the DNA. As you can see, it is shaped like a double helix. In between each one of those round shapes are the 4 nitrogenous base pairs. Each base is always paired with the exact same base. I'm now going to draw a model of the RNA. As you can see, it has only one strand. It only contains one base of the base pair. Now we're going to move on to the 2 types of RNA involved in protein synthesis. There's the messenger RNA and transfer RNA. They each have their unique role to play in protein synthesis. The role of messenger RNA is to copy the coded message from the DNA in the nucleus, then carry it to the ribosome in the cytoplasm. Transfer RNA's role is to carry the amino acids to the ribosome and then add them to the growing protein chain. Now on to the 4 steps in protein synthesis. The first step is for the DNA molecule to unzip between its base pairs. So, instead of 2 strands, you now have 2 separate strands with the base pairs split. Now, one strand of the split DNA will direct the production of the messenger RNA. Soon, the RNA bases will pair up with the DNA bases. On to step 2. Messenger RNA will now leave the nucleus and it will enter the cytoplasm where it can attach to a ribosome. On the ribosome, the messenger RNA will provide the code for the protein molecule. During this time, the ribosome moves along the RNA strand, messenger RNA strand. On to step 3. The transfer RNA will now attach to the messenger RNA. The transfer RNA reads the message by pairing up its 3-letter codes to the bases on the messenger RNA. The transfer RNA carries amino acids that link in a chain. This order of the amino acids will be determined by the order of the 3-letter codes on the messenger RNA. Now on to the final step. In this step, the protein molecule will grow longer as each transfer RNA puts the amino acid along the growing protein chain. Once the amino acid is added to the protein chain, the transfer RNA will be released into the cytoplasm where it can pick up another amino acid. Each transfer RNA always picks up the same kind of amino acid. And the process continues. Now on to mutations. They can occur in a gene. There are 3 types of mutations that can occur in a gene. There is addition, substitution, and deletion. Addition is when an extra base pair is added to the gene. Substitution is when a base pair is cancelled for another. Say you had adenine and thymine base pairs. Instead of that, cytosine and guanine would be added. Deletion is when a base pair is cancelled from the gene. What are the effects of mutations? Are they good, are they bad, or are they neither? They can be all 3. A good mutation would be a bacteria that is resistant to an antibiotic. An effective bad mutation would be like Down syndrome. What would be a not good or bad mutation? Say there was a white lemur that should be brown that's living in the zoo. There would obviously be no harm. Thank you guys for watching. I hope you learned something new. My inspiration credits go to Tanvi Gujral.